Okay, we should be live now. So I'm just gonna wait until it actually catches up. But yeah, I think we're live now. Welcome everybody. I'm Michael, and today we're hosting Professor Julie Semmelhack from the Hong Kong University of Science and Technology. Yeah, she's gonna talk about zebrafish and some behavioral state switches. We're really excited. And take it away, Julie.
Okay, let me share my screen. Okay, can you guys see the screen? Okay, so great. So today I'm going to talk about a bit about a published story and have time, hopefully, for a couple of new stories on stimulus identification and behavioral state transition. And so animals perform a wide range of innate visual behaviors. And so here, hopefully in this video, you can see this bird is performing a freezing behavior. And so this is an immobility behavior to avoid detection by predators that can be triggered by visual stimuli. But then if the predator gets too close, eventually the bird will change strategies and do an escape when the threat is more imminent. And then the third visual behavior in this video is, of course, hunting, which is being done by the cat. So we still don't know very much about how animals actually switch between these behaviors and how they identify objects as either prey or predators. And so we're interested broadly in, first of all, how you go from stimulus to behavior. So how you identify the stimuli, how you decide to perform a behavior, and then how this pathway is affected by things like internal state or environmental context. And so we study this in using the visual system, and visual stimuli, even simple visual stimuli, can evoke a variety of behaviors. So for example, small moving dots can evoke hunting in zebrafish. And then in many different animals, predator-like stimulus, if it's a constant size, will tend to evoke freezing. And then a looming stimulus will tend to evoke escape. And so we study this primarily in zebrafish, both because of the optical clarity of the larval fish, and this allows us to do functional imaging and optogenetics in the intact organism. And then it's relatively simple brain with roughly 180,000 neurons. And then also the fact that even as larvae, they will perform a variety of visual behaviors. And we can study this in a head-fixed preparation. So basically what we do is we take the larva and we embed it in a drop of agarose. And then we can cut away the agarose around the eyes and tail so that we allow the eyes and tail to move, which is how we read out what they're trying to do behaviorally. And in this head-fixed preparation, they still perform a number of visual behaviors. And one of those would be prey capture. And so here's an example of a embedded larva doing prey capture. So on the top, you see the visual stimulus, which is going to be a UV dot on a red background. And so when the stimulus comes on, you'll see the eyes converge and the tail start to do orienting swim towards the prey. And so it turns out that UV is the best stimulus for this. And so we use a light crafter projector, and we showed previously that the UV is the optimal stimulus compared to the two other colors. And for anyone who's interested in it, we have a protocol describing how to set this up, which I would highly recommend for anyone studying prey capture. And so that's the hunting behavior. And then, as I mentioned, many other systems have these two kinds of defensive behaviors, escape and freezing. So everyone from flies to mice to humans. And previously in zebrafish, we had described an escape behavior. So if you have an expanding dark stimulus, they will do this big escape turn. However, previously visually evoked freezing hadn't been described. And so my students, Fred and Yushin, wanted to see if they could study to get this, this behavior working. And so this is our setup. So we basically have a head-fixed zebrafish in this on a cover slip, and it's in this behavioral chamber, and one side of which is a screen, and then we project the visual stimulus on the screen, and at the same time we record the behavior from above and also from the side. And so when we show a dark stimulus of constant size, which we call a sweep stimulus, as opposed to looming, one thing we could see in a fish that was doing spontaneous swimming, when the stimulus comes on in a minute, oftentimes they will stop swimming, and then this swimming behavior resumed after the offset. And however, and so we can quantify this, so the probability of swimming goes down during the stimulus if it's this predator-like sweep stimulus. However, not every head-fixed fish is going to be spontaneously swimming. So we wanted a second way to have a readout of freezing, and so we also looked at the heart rate. And so bradycardia in many systems is a signature of freezing because freezing is an output of the parasympathetic nervous system, and one of the outputs is to the heart. And so we'll have a transient decrease in heart rate at the start of the stimulus. And so we can very easily record the heartbeat in zebrafish larvae. And so what you can hopefully see is at the onset of the stimulus, the heart kind of skips a beat. And as you can see here in the heart rate per minute readout. And so we quantify that by taking, identifying all the rhythmic pixels in the heart region, and just taking the pixel intensity, you can see the heartbeat already in the peaks. And then we can quantify the change in heart rate during the stimulus period. And so here we see in the case where we added the freezing or sweep stimulus, we saw this transient decrease in heart rate at the onset of the stimulus compared to the control. And so we decided to use this stimulus to as our readout for freezing. And over the population, we can see that there's a significant heart rate change during this sweep stimulus. And it turns out that the best stimulus for evoking freezing is about a 15-degree visual field diameter dark stimulus. So that was our standard sweep stimulus. And so we wanted to know, first of all, which neurons actually respond to this stimulus within the visual system, which are possibly detecting it, and then which neurons are responding when the larva actually freezes. And so the kind of prime suspect would be the optic tectum, which is the largest visual area in zebrafish. And so we hypothesized we might find some sensory neurons that receive retinal input and just detect the visual stimulus. We were also interested in whether there could be some neurons in the optic tectum or elsewhere that actually respond when the fish decides to freeze, and whether we could ask, is that where the decision actually takes place? And then if there's some kind of behavioral state, input to the optic tectum that could affect this behavior. And then finally, how does this pathway interact with the other ones for visual behaviors such as hunting and escape? And so to look at this, we designed a battery of stimuli, including 3 behaviorally relevant stimuli. So the sweep stimulus, which triggers primarily freezing, the small UV dot stimulus, which triggers hunting, and then the looming stimulus, which is an expanding dark object that triggers escape. And so we presented all of these in a screen in front of the larva, and at the same time recorded their behavior and imaged their brain. And so all the larvae in the data set had to respond with all 3 behaviors at least once. And so we basically use a similar setup to record behavior under the 2 photon, where we present the stimulus in front of the larva, and then we record their behavior, so we can record their heart as well as their eye and tail movements. And then for this data set, we wanted to image a relatively large volume covering most of the areas that could be involved in detecting the visual stimulus and mediating the behavior. And for this, we expressed GCAM6 as panoronally, and we did 2-photon imaging using an electrically tunable lens so we can quickly switch between planes. And we imaged 14 planes covering this portion of the brain. And so each of these gray dots represents one cell in our data set. And we mapped the locations of all the cell bodies onto the MAPC brain atlas. And so this in total is data from 7 fish. And so some of the areas we covered include the optic tectum, the pretectum, thalamus, pallium, and nucleus isthmi. And in total from this data set, we segmented 180-something thousand cell bodies. And then the next question, of course, was how to analyze the data. And so we first wanted to identify what we were thinking of as sensory neurons, or basically neurons that kind of detect the visual stimulus. And we defined these based on the principle that they should be highly periodic. And so what we did was to look at the trace of a single neuron. And so this is a neuron responding during 8 sweep stimulus trials, which are the pink regions. And so we first calculated the average response of that neuron and then concatenated that to get trace average. And then we compared the variability of the real trace versus the trace average. And so in principle, a perfect sensory neuron should have the same variability in these two. So we use this to calculate a sensory index. So neurons with a high sensory index should be highly periodic. And we see that neurons in the top 1% of the sensory index indeed respond quite reliably to the visual stimulus, in this case, the sweep. And so we first had to use a regressor a bit. So we took the top 10% stimulus correlated neurons and then used the SI to take the top 15%, and then did spatial p-value filtering to identify neurons that were co-localized across multiple animals. And so using this, we got a population of sensory neurons. So each neuron has a sensory index for each stimulus, so 3 sensory indices. And we, using this threshold, we identified sensory neurons for all 3 stimuli. And then we looked at where they were in the brain. And so as you can see, so here we're plotting the number of neurons in each brain area. And so the largest number of all 3 classes were in the optic tectum, as you can see here. And then we also saw a quite large population of prey sensory neurons in the pretectum, which is consistent with previous findings. And we looked a bit at the functional properties of these populations. So, so this is the delta F over F average response of the whole population of sweep sensory neurons to the 3 stimuli. And so as you can see, the sweep sensory neurons respond quite well to the sweep stimulus. Interestingly, they also respond to the prey stimulus. And similarly, the prey sweep sensory neurons also respond a bit to the sweep stimulus. And we also looked a bit at the individual neuron responses, so their functional properties. And so here we're plotting each individual neuron's sensory index. So in blue, we have the prey sensory neuron. And here we're plotting their prey sensory index versus their sweep sensory index. So by definition, they all have a high prey sensory index, but many of them also have a quite high sweep sensory index, meaning they responded quite reliably to the sweep stimulus as well. And many of the neurons were actually classified as both prey and sweep sensory neurons. So there seems to be quite a bit of overlap between these populations. And then we looking at the individual neuron responses, we can see, for example, this example prey sensory neuron shown in G also responds quite well in the sweep stimuli trials shown in pink. And the example sweep sensory neuron H responds also fairly well to the prey stimulus. We also did this analysis for looming and sweep sensory neurons, and we also saw some overlap between the 2 sensory populations there. And so we next wanted to look at the behavior correlated neurons. And so at the same time we were doing the imaging, we were recording these three behaviors. And so first of all, we have the looming behavior, looming stimulus, which evokes escape. I already showed you the sweep stimulus, which evokes freezing, and as read out by the bradycardia. And then we have the small UV stimulus, which evokes hunting, and that we primarily read out by eye convergence. And so we recorded these three behaviors. In this imaging data set, we did indeed see that the sweep stimulus primarily gives us a freezing, also a bit of escape in the green here. The looming stimulus primarily gives escape responses, also a bit of freezing. And then the hunting stimulus, the prey stimulus, almost entirely hunting. And so then we wanted to identify neurons that responded when the animal actually decided to perform these behaviors. And so to do that, we wanted to do that somewhat quantitatively, but for example, this is looking at one brain region, the nucleus isthmi. We can see that when we present the sweep stimulus, we see some neurons are activated, but then in sweep stimulus trials where the animal actually had bradycardia, there was a larger population of active neurons. And then in the same brain area, when we presented the escape stimulus, we saw basically no response, but when the animal actually did escape, we saw a different population of neurons light up. And then when we presented the prey stimulus and then they decided to hunt, we saw a third population. And so to more rigorously quantify this, we developed an approach to identify what we're calling sensory motor neurons that are correlated with each of the three behaviors. And so instead of using a motor regressor type of approach, we decided to try to kind of decompose the activity of each neuron into the sensory and behavioral components to avoid the issue of correlation of the sensory stimulus with the behavior. And so basically what we did for each neuron's trace, we took the average and then we subtracted that trace average from the original trace to get what we call the trace surplus. And then as you can see, sometimes there's a little bit of an extra response in this trace surplus. And then we ask how well that is correlated with a regressor for the behavior. So in this case, if the fish froze in the trial 2 and trial 6, we would have this kind of regressor, which would be well correlated with this trace surplus. And we use that correlation to calculate a score. And so we call that the MSI, or motor surplus index. And we can see that neurons with a very high MSI tend to be highly correlated with the behavior. So for example, this one responded just in the freezing trial, which was here. And so we use this MSI to set a threshold for what we call sensory motor neurons for each of the three behaviors. And then we use spatial p-value filtering and got a set of sensory motor neurons for freezing, hunting, and escape. And so the functional properties of the sensory motor populations, basically unlike the sensory populations, the sensory motor ones tend to respond primarily when the fish actually behaves. So there's very little response in the no response trials. And that's true of freezing, escape, and hunting sensory motor neurons. And these populations are quite separate between the sensory and sensory motor populations. And then where these neurons are within the whole imaging volume. So here we're showing the location of freezing in red, escape in green, and hunting in blue sensory motor neurons. And you can see that there is some irregularity in their distribution. And some of the kind of interesting areas are, so for one thing, that we have all three classes in the optic tectum, not too surprisingly. And then some other interesting areas are the thalamus and pallium. And so if we zoom in on these, you can see that there seems to be kind of a region of the medial thalamus that has more defensive correlated sensory motor neurons. And so there seems to be some anatomical separation there. Interestingly, in the pallium, which is the homologue of this is the dorsal medial pallium, which is the homologue of the amygdala, we see only freezing and escape sensory motor neurons, no hunting. And suggesting that this could be the homologous region in the fish. And then finally, we looked a bit at the functional properties of these classes. And so one interesting thing is that the populations are fairly specific. So the sensory motor neurons for freezing tend to just respond in freezing behavior, not so much even in escape, for example. And the population of freezing sensory motor neurons had very little overlap with the population of escape or hunting sensory motor neurons, suggesting that once you decide to do one of these behaviors, the pathways are kind of segregated. And so to summarize kind of the position of these neurons, we found that the optic tectum has sensory neurons for all three classes, and also sensory motor neurons for the three behaviors. And I'm not going to talk about this today, but we also did some correlation analysis to show that these sensory and sensory motor populations within the tectum are likely connected, suggesting that these sensory motor neurons are at least integrating sensory information. And then we also saw several other regions, such as the pallium, thalamus, pretectum, and nucleus isthmi with sensory motor neurons, consistent with other systems, suggesting that we can use this to study these kind of ancient pathways for highly conserved visual behaviors. And this work was published a couple of months ago, so I only talked about a very small portion of it, so please feel free to check it out if you are interested in further details. There's a lot more in there. And okay, so then I'll switch gears and talk a bit about some new stuff that we're doing more recently. And so one kind of remaining question from this work was, how do animals actually identify objects as prey and predators? So how do they identify this UV object as something that you should hunt versus this sweeping stimulus as a potential predator versus this looming stimulus as an approaching predator? And and one interesting thing is that we do have multiple outputs of the retinal ganglion cells to different areas. So retinal ganglion cells project to the optic tectum, pretectum, and thalamus. And we don't really know what the point is of the different projections to the pretectum and thalamus. This is something I've been wondering about actually since grad school, which I recently realized that I did write my postdoctoral fellowship on this very question. And so we're finally starting to get some traction on it, hopefully. And then in my postdoc, I worked on this area called AF7, which is prey responsive. And interestingly, the retinal ganglion cells that project to AF7 also project to the optic tectum. They have a collateral in AF7 and the optic tectum. So there was always the question of why do you need two areas that respond to prey? And the same is true of the thalamus. So we have retinal ganglion cells that arborize in the thalamus and then go on to project to the optic tectum. So why do you need to send the information to two places? And one kind of simple hypothesis could be that basically the optic tectum is, a map of visual space that's identifying where the stimuli are. And then these other areas like the pretectum and thalamus are actually identifying what kind of stimulus the object is. And so we wanted to investigate this in larval zebrafish a little bit more because in our previous data set, we only had 3 different stimuli. We didn't really play with the visual parameters of the stimuli at all. So we designed this larger stimulus battery for sensory profiling. And so we created a total of 40 visual stimuli, varying different features. So we varied the size of the stimulus. We did prey stimuli as well as sweep stimuli. We varied the color, so UV versus red and on versus off. And we did some global stimuli such as whole field luminance changes and then dark and bright flashes. We varied the elevation and then the direction of the stimulus. And this is kind of a pilot data set that we just started analyzing recently. So this is kind of hot off the presses. And so we did a similar recording setup. So we expressed GCamp panoramally. We used an ETL to record 15 planes. And we collected a data set of 11 fish, each of which have about 40,000 cells segmented. And then, and so this is our data set. We actually recorded a bit more caudally. So in this data set, we are not recording the forebrain, but we are going further back into the hindbrain. And we're still hitting the optic tectum, pretectum, thalamus, and nucleus isthmi, as well as cerebellum and medulla. And so as a kind of first pass at analyzing all this data, we recently just looked at the sensory neurons. So to see if we could break down the sensory neurons into neurons that respond to different stimulus features. And so again, we just look at the variability of this, trace versus the average trace to get the sensory index. So we find these highly periodic sensory neurons. And then we clustered those neurons. And so, so we've just started looking at this data. So the first thing we did was to do a raster map of these sensory neurons. And so when we got 15 clusters, and so we see some kind of interesting features. And so, so first I'll highlight cluster 1, and zooming in on cluster 1, you can kind of see, so here we have the different size stimuli that we presented from 2 to 12 degrees of the visual field. And so these sensory neurons in this cluster tended to prefer the small size stimulus. They also preferred, for the color part, they preferred a bit the on stimuli, the UV or red. And then we can plot the responses over time to the different kinds of stimuli. And so for cluster 1, for the size stimuli, we can see that the 2-degree stimulus gave us the best response. And then these cells responded quite well to UV, UV on red or UV on dark, or red on dark, not so well to the dark stimulus in magenta here. And then they responded best to the praise stimulus, a bit to the sweep as well, but not at all to looming. And then in terms of the position of the visual field that triggered the response, we saw basically praise stimuli on the contralateral side triggered quite a robust response. Interestingly, prey on the ipsilateral side also triggered a response. So these cells are kind of binocular. And so they seem to not really care where in the visual field the prey stimulus is. And then, so just zooming in on one of the other interesting clusters that we saw, so this is cluster 3. And so basically this cluster tended to prefer larger stimuli. And so if we look at the size response for prey, it preferred the 12-degree stimulus, actually responded to all of the different color on-off, both fine, responded best to sweep and looming, and then it was completely monocular, so didn't respond at all to the ipsilateral stimuli. And so maybe a bit more spatially restricted. And so, so that's kind of interesting, but then of course we would like to know what are these clusters actually doing, and are they actually important for the behavior? And so we already have some clues to this. And so the way we did that was, so as I told you before, praise stimulus gets us hunting, sweep stimulus gets us freezing. And this is defined by the eye angle for hunting and the bradycardia for freezing. And so what we found that we could do was actually add in a competing stimulus to the hunting paradigm. So if we present the prey stimulus, the same prey stimulus, but we add a sweep stimulus on the other side, we can actually cancel out the hunting. And so in this case, most of the time, the fish will freeze. And so these behaviors are mutually exclusive. You can't do both at the same time. And so you just see the bradycardia and you have immobility, and you don't see the eye convergence. So basically freezing tends to win. It's probably the sweep stimulus is more salient. And so what we can do is have exactly the same prey sensory stimulus, but then manipulate the behavior such that they now don't do the hunting. And then we can ask what happens to the responses of the neurons that responded to this hunting stimulus. Presumably, assuming that not everyone gets suppressed, the ones that are suppressed are probably playing some important role in identifying the stimulus as prey or mediating the behavior. And so we looked at what happened to the prey sensory neurons in this competition experiment. So first of all, just the behavioral as a population. So hunting, so the prey stimulus gives us mostly hunting, sweep stimulus gives us mostly freezing, and then what we call the competition stimulus, which is both sweep and prey, gives us basically mostly the same degree of freezing and a little bit of hunting. And so if we break this down by different fish, so each bar represents the responses of one fish. And so you can see that most of the fish in the competition trials, they were mostly freezing, and very few were hunting, except for this one very brave fish that still kept on hunting. And then we can look at the suppression of the prey sensory neurons with the addition of that competing sweep stimulus. And so here we're plotting the delta F over F of each of the prey sensory neurons in the prey trials versus in the competition trials. And so you can see that some of the neurons are not really affected by the addition of the sweep stimulus, but some neurons down here are strongly suppressed in the competition trials. And so we can then calculate what we call a prey suppression index, which is the response in the prey only condition minus the competition over the total. So a prey suppression index of 1 would mean it's totally suppressed, and 0 would mean it's not suppressed at all. And so when we do this for those two clusters, what we see is that, so cluster 1, which was kind of a prey preferring cluster, was highly suppressed. And so that's shown in orange here. So the prey suppression index of the neurons in cluster 1 were kind of ranging from somewhat suppressed to totally suppressed over here at 1. Whereas cluster 3, which was the dark moving object preferring cluster, basically did not have any suppression with the addition of the competing sweep stimulus. And so the average of cluster 3 was somewhere around 0. And that's similar to all the prey sensory neuron average prey suppression index, suggesting that this cluster 1 is really kind of an outlier and could be involved in recognizing the prey stimulus and is suppressed when we add in the sweep stimulus and the fish decides not to hunt anymore. And so, and then where in the brain is this cluster? So cluster 1, the prey, what we're thinking of as the prey ID cluster is mostly in the pretectum. So this is just the number of neurons. So the largest population was in the pretectum, and a few in the optic tectum. And then cluster 3 was predominantly in the optic tectum, as you can see here. And so this is pretty preliminary, but then in terms of what is actually mediating the suppression, the most likely candidate is the nucleus isthmi, which is an accessory nucleus of the tectum, and it's been described in classic work by Eric Knudsen in birds as providing global inhibition or local excitation to the tectum. And Miguel Fernandez in the Bayer lab also looked at its influence on the tectum in zebrafish. However, the nucleus isthmi does have some lesser known projections to the pretectum and thalamus, which could be mediating perhaps the inhibition of the prey sensory neurons that we're seeing. And so that's where we are for now. We're still working on the analysis of this data set. So our next steps are clustering all the neurons to get a better idea of the whole population. And we'll see if we can find any predator ID clusters. And then we're planning to collect a new data set. And some of the directions we want to go in are getting better spatially restricted stimuli so we can really ask whether the optic tectum receptive fields are indeed smaller than the pretectum and thalamus, and better evidence for that kind of where versus what hypothesis, and then possibly manipulation of the pretectum and thalamus to test if they're actually involved in encoding the identity of the stimulus versus the tectum doing the location. And that's the sensory stimulus identification side of the new stuff. And the other new questions we're looking at are how these innate behaviors are modulated by internal states. And so this is obviously a big question in neuroscience. So animals in the presence of hunger or fear are going to modulate their response to food or predator stimuli. And how this happens is relatively unknown. And in zebrafish larvae, there's been some descriptions of some internal states. And one cool paper was from the Rowley lab a couple of years ago, in which they looked at internal state transitions in freely swimming fish and found these two states, which they called exploration, which was where they would travel a larger distance, versus exploitation, where they stayed confined to a relatively small area. And they would switch spontaneously between these two states. And so in our head-fixed fish, we do see a lot of variability in the behaviors. So this is each dot here represents the behavior of one fish. So you can see that there are some fish that do quite different proportions of behaviors. So for example, if we look at this red line, this is a single fish that responded quite a bit to the sweep stimulus with freezing and not at all with escape. And then another fish in the same data set responded predominantly with escape to the same sweep stimulus. So there is quite a bit of individual variability. And so we wondered if some of it could be due to the fish being in a different internal state. And another question is whether the head-fixed fish, we can actually observe these two types of states in head-fixed fish. And it turns out that we can, but it's quite temperature dependent. So this only really happens if you heat them up to 28 degrees. Otherwise, most of the head-fixed fish will spend most of their time just sitting there. But if you heat them up to like proper physiological temperature, we can see them do quite a few spontaneous swims. And so here's one example. So during this time period, you can see the fish was doing quite regular swimming. And this might last for a minute. And then it entered a more passive or quiescent period where it would spend a minute without doing any swims. And so as you can see over here, it's completely still for a minute. And so we wanted to know then, can we quantify these two states? And so to do this, we use the tail angle. And let me see if I can start this video. So we basically use the swimming to infer the distance that the fish were intending to go. And so a bigger tail angle would correlate with a larger distance intended based on the free swimming fish. And so we can calculate the head-fixed larva's intended displacement based on their tail movement. And so we can use that to generate this plot of intended swim distance. And so we can see that there's kind of a distribution of short versus long periods of swimming. And so we use this to infer two states, which we're calling preliminarily passive and exploration states. And so typically when they're in the no swimming or short distance swimming condition, they're in the passive state. And when they're doing more long distance swims, they're in the exploration state. And so we can use this model to classify our behavioral data as either being passive or exploration. And so then the next thing we wanted to ask is, does this, what we're calling passive and exploration state, actually modulate visual behaviors? And so we have the prey stimulus and the sweep stimulus. We also designed what we call an ambiguous stimulus, which is kind of midway between the prey and sweep. So it's a smaller dark dot. And that one will trigger both hunting and freezing. And so we then use these visual stimuli to ask what is the response when the larvae are in the different states. And so for this experiment, we looked at the swimming behavior before the visual stimulus, and then put my pointer back, and then we presented the visual stimulus over here, and we used this pre-stimulus region to classify the state as either passive or exploration. And so we did this for all three of these visual stimuli. And so, okay, and so this is still quite preliminary. So we have a data set of 14 fish. And what we saw basically was that when the fish were in the passive state, in response to the ambiguous stimulus, they tend to be more likely to freeze. When they're in the exploration state, the chances of freezing are much lower. So that is the difference in the amount of freezing is significant there. And also in the passive state, they're more likely to freeze in response to the looming stimulus, as you can see here. And so it seems like basically if they're in the passive state, no matter what, they're more biased towards freezing, regardless of whether it's the looming stimulus or a ambiguous stimulus, or even sometimes the prey stimulus. And so suggesting that these passive versus exploration states are really modulating visual behaviors. And then next steps, we're planning to, first of all, see if we can do functional imaging during these state transitions and identify which brain areas might be triggering the transition. And then we would, of course, like to see how the transitions affect visual circuits. So at what point are we seeing modulation? Is it in the sensory neurons, sensory motor neurons, tectum, hindbrain, or where? And so, yeah, that's kind of our future plans. And so finally, I'd like to thank my lab, particularly Fred and Yuxin, who did a lot of the work, and as well as the rest of the lab, my collaborators and funding sources. And thank you for your attention, and I'll be happy to take questions.
Great, thanks. Really cool talk. So for the audience, if you post the questions in chat, then I will ask them further to Julie. And also, as I posted in chat, after we're done with questions, I will open the Zoom to the public. I'll put the link in the chat and feel free to join and maybe directly talk. So currently there's no questions in chat, but I have some. Most of those you actually answered when I wrote them down, and then the next slide explained it already. But one thing I was wondering is if you can do the reverse. So you have the internal state influence how the stimulus reacts to a stimulus. But for example, if you show them a bunch of predator stimuli, do you think that would lead to a state change, maybe?
Yeah, so there is that paper by Herman Sumbray's lab with kind of a state change after like escape. So, and we do see that basically when we show like a looming stimulus and or a sweep stimulus and they decide to escape, then there's typically like a stationary period after that. So they have a decreased likelihood of spontaneous swimming for 30 seconds.
Okay.
Yeah, so there may be some kind of change in the swimming probability after the escape. Yeah, but so far since we typically don't show another stimulus in that time period, so we don't know if they are responding differently. Yeah, good question.
Okay, thank you. Then Tom, first he says great talk, and then Did I catch that right, that they hunt more in the exploration state? Is that in line with the idea of exploration versus exploitation? I noticed that you call it passive state, not exploitation.
Yes, so we are calling it, we're not calling it exploitation because we don't see an increase in hunting. Yeah, so basically there was no effect on hunting. And so they will hunt in either state. So it's not exactly the same as the Rolly Lab exploitation state, I would say. Yeah.
Okay. Kit Longton also says, great talk. Currently no other questions, then I will ask another one. So in the, hold on, which one? So in the first part, you kind of group pretectum and thalamus in the part where you figure out which neurons have which the most preference and how that changes between brain areas. Did you look within pretectum, like within individual arborization fields as well, or just grouping it at the moment?
Yeah, I mean we're basically we're not really breaking it down by arborization fields,
Mm.
just regions so far based on the MAPC Brain Atlas. Yeah, so we
Okay.
can't say whether these neurons are getting input from a certain AF, for example, just what region they're in.
Okay.
Yeah.
I'm guessing that in this case you have a pan-neuronal line, which I also have in the frogs, but in our case it's not nuclear localized. I guess you have nuclear localization.
Yes. Yeah.
Okay, yeah,
Yeah.
that
So
makes
that
sense.
just makes it a lot easier to do large scale imaging and the segmentation for us.
Okay, yeah, that makes sense. Um, I just posted the, I think I just posted the link to the Slack, to the Zoom in the Slack. So if you want to join, come in. Tom asks okay, there's more questions coming in. Tom asks, I probably missed it, but did you see a difference in brain/behavior if you used red UV or black dark dots? Also, did you ever try white?
Hmm. Uh, so difference in behavior for yeah, so hunting, UV is going to trigger the most hunting. In terms of behavior, they sometimes also freeze to the bright dots, but not as reliably. So that's behavior. And then yeah, I guess neuronal responses, we're just starting to get into characterizing them. But there seems to be things that respond to everything. So yeah, it's a bit complicated at the moment.
Uh, then another question from Panky. Did cluster 3 neurons show no suppression?
Yeah, so
Are they on the contralateral side of the prey or the sweep?
they are on the prey side and they showed no suppression. So I kind of thought, you know, maybe everything on the same hemisphere will be suppressed, but that's not the case. So these are neurons on the prey side that are not at all suppressed. So I think their index was like quite close to 0. Yeah, yeah. So the cluster 3 was basically centered close to 0. So uh, hardly at all suppressed. Yeah, and they were on the prey side. So there does seem to be, there might be some specificity. We have to look more, and I think we have some other pretectal clusters that are not suppressed. So it's not just that everything in the pretectum is suppressed or everything on the same side is suppressed. Yeah.
They have a follow-up question. To what extent the cluster 1 sensory response correlates with hunting in the sensory profiling test?
Yeah, I think we haven't analyzed our sensory profiling data for behavior,
Mhm.
but we were using quite a short intertrial interval to try to get in more stimuli. So because we have 40 stimuli and 3 trials each. So in order to get it all done in a reasonable time, we had to shorten the interval, which means they don't behave as much. And so we don't know. I don't know if they behave. I think so. You're asking if they were correlated with hunting, I think. And I think cluster one had a little bit of it was a sensory. They are sensory neurons. I think there is a very slight enhancement in hunting trials. Yeah.
Okay. Cool. I think that's it for questions. I'm going to post the Zoom link one more time. Feel free to join. Otherwise, thanks a lot for the talk. It was really cool. Great to host you. Yeah, and then I guess we can just wait a bit if people want to join. I will have to leave the live stream open for a bit for that. Otherwise, the link will also disappear. So maybe let's give a couple of minutes for that.
Okay, cool.
Great. And then maybe you can also end the slide screen share thingy. Yeah, because I think that's currently still on. Okay. Good. So this part is also still live streamed, just as an FYI. I will let you know when I kill the stream. Anyone who wants to join within the next like 1 or 2 minutes, please.
Oof, now I'm live. That was great.
Yep.
Lots of really interesting stuff. I'm going to ask you lots of questions about how to keep the behavioral state happy in a minute.
Okay.
Yeah, that was really cool. Also the like in the identification part. Yeah, I have some questions, but they're more like open-ended, where like it's hard to say if that actually makes sense or not. Like for example, in the competing stimulus part, basically you said that it might be the nucleus istemi. Do you think that would also be involved in like modifying stimulus responses in the internal state dependent thing, or could that be a different mechanism?
I think internal state, probably different area. So the nucleus istemi is probably more kind of stimulus like saliency and competition, and then internal state. Yeah, it could be so dorsal raphe nucleus, hypothalamus, who knows, like these kind of more state related areas would be the top candidates. Yeah.
Okay, cool. So then I'm gonna cut the stream here. One last time, thanks a lot, and see everyone for the next stream. Okay, and we are offline.